Welcome back, my dear students. Today we are going to discuss one of the most fascinating and important topics in biology, the cell membrane, also called as the plasma membrane. Have you ever wondered how a tiny cell manages to control what enters and what leaves, just like a security gate? The plasma membrane does exactly that. It's not just a simple covering, it's a selectively permeable, highly dynamic structure that maintains the life of the cell. In this video, we will delve deep into its structure, including the fluid mosaic model, and explore its function like transport, communication, and signaling. By the end, you will clearly understand why the plasma membrane is called the gatekeeper of the cell. So let's begin our detailed journey into the cell membrane structure and function. Walter Gilbert, this is the image of Walter Gilbert. He proposed the RNA world hypothesis in 1986, suggesting that around 3.8 billion years ago, when life first originated on Earth, it was the RNA molecule that played a central role. RNA, capable of both catalyzing chemical reactions and storing genetic information, is thought to have been the first self-replicating molecule. Over time, this RNA molecule likely developed a membrane around it, giving rise to the first prokaryotic cell. Thus, it can be concluded that the cell membrane evolved around the same time as the origin of life, approximately 3.8 billion years ago. The cell membrane is therefore essential to the life of the cell. We know the cell membrane plays a crucial role in maintaining organization and functionality. It provides compartmentalization, ensuring the segregation of different cellular activities and organelles, thus promoting efficiency. It serves as a site for biochemical activities by hosting enzymes and facilitating vital metabolic processes. Its selectable permeable nature allows the regulation of molecular passage, presenting only specific substances to enter or exit the cell. The membrane facilitates the transport of solutes, maintaining the homeostasis by actively or passively moving substances like ions and nutrients. It enables the cell to respond to external stimuli, acting as a sensory interface with the environment. Furthermore, it promotes intracellular interaction, supporting communication and coordination among cellular components. Lastly, the cell membrane plays a pivotal role in energy transduction by participating in processes like ATP synthesis, which is crucial for cellular energy supply. This is the diagram showing the basic structure of the cell membrane. Brief history of cell membrane study. In 1855, Carl Nageli and August Kramer were the first to observe the cell membrane. The lipid monolayer model proposed by Irving Langmuir in 1881 to 1957, his year of birth and year of death, marked the first scientific attempt to understand the structure of the cell membrane. Langmuir suggested that the membrane consisted of a single layer of phospholipid molecules, with each molecule arranged one molecule thick. In 1902, Ernest Overton from Zurich University proposed that the cell membrane is composed primarily of lipids. In 1925, Dutch scientists Gorter and Grendel proposed that cell membrane is a lipid bilayer. They hypothesized that if the plasma membrane were indeed a bilayer, the surface area of the lipid monolayer would be approximately double the surface area of the plasma membrane or cell membrane. To test this hypothesis, they conducted an experiment in which they extracted lipids from a known number of red blood cells or erythrocytes from various mammalian sources, including humans, goats, and sheep. They then spread the extracted lipids as a monolayer and measured the total surface area of the plasma membrane of the red blood cells. Using Langmuir's technique, they also measured the area of the lipid monolayer. By comparing the two surface areas, they calculated an estimated ratio of 2:1, supporting the idea that the plasma membrane is a lipid bilayer. In 1931, American biologist James R. Ploy named it as cell membrane. In 1935, James Danieli and Hugh Dabson proposed the lamellar or sandwich model for the cell membrane. According to their model, the membrane consists of two layers of proteins positioned on both sides of the lipid bilayer. This is the image showing the sandwich or Dabson-Danieli model of cell membrane. Here in the structure, there are two layers of proteins. The green is the layer of protein. There are two layers of protein positioned on both sides of the lipid bilayer. Lipid bilayer both sides, there are two layers of proteins. The protein layers are the hydrophilic zone and the lipid layers is the hydrophobic zone. In 1959, J.D. Robertson proposed the unit membrane model. This is the image showing the unit membrane model, also known as PLP model or phospholipid protein phospholipid model, based on observation under the electron microscope. Surface tension measurements taken on the membranes suggested the presence of protein, supporting the idea of a protein-lipid protein arrangement in the membrane structure. Now we should study about the fluid mosaic model. In 1972, S. J. Singer and G. Nicolson or Gert Nicolson stated fluid mosaic model, which is called as the central dogma of membrane biology. Basically, this model postulates that lipid and integrated proteins are disposed in a sort of mosaic pattern, and all the biological membranes have a quasi-fluid structure where both lipid and protein components are able to perform transitional movement within lipid bilayer. So we can say that cell membrane is also called as plasma membrane. It's roughly its size near about 5 to 10 nanometer. Average is 7 nanometer. Semi cell membrane is also it is semi-permeable nature because it allow the entry of certain particles, not all of them. So it is called as semi-permeable. It is present around every living cell. Cell membrane is the outermost covering in all animal cell. In case of plant cells or bacterial cells, there is cell wall. But just beneath the cell wall, in plants and in bacteria, there is the cell membrane. But in animal cell, there is no cell wall. So in case of animal cell, cell membrane is the outermost layer. The main functions of the cell membrane are the maintain the cell shape and support the cell. It is responsible for selective transport of materials. Structure and composition of cell membrane. Cell membrane basically consist of membrane lipids, membrane proteins, and glycolipids and glycoproteins. In 1972, Singer-Nicolson proposed fluid mosaic model on the structure or function of cell membrane. According to the fluid mosaic model, cell membrane is fluid in nature. So what do you mean by fluid? Because their constituent parts, their constituent molecules flow within the structure. So it is called as fluid. And why it is called as mosaic? Because there is asymmetric arrangement of different molecules. There are different types of proteins or different types of lipid molecules, but their arrangement is not fixed anywhere throughout the structure of the cell membrane. So membrane lipids, there are basically two types of membrane lipids, phospholipids and cholesterol. And we know the fluidity of the membrane is influenced by the temperature. In higher temperature, the membrane fluidity increases, and in lower temperature, membrane fluidity decreases. Fatty acid we know unsaturated fatty acid and saturated fatty acid. If there is more unsaturation, then we can find the kink formation in the phospholipid molecule structure, and it will increase the fluidity of the membrane. But as much as saturated fatty acids are there, the fluidity of the membrane will be decreased. So cholesterol maintains the fluidity. We know at high temperature it maintains the fluidity. We know at high temperature normally phospholipids become more fluid. So cell membrane becomes more, increases their fluidity. But if cholesterol molecule is there, it will restrict the movement of the phospholipid molecules. So it will in the higher temperature, when the mobility of the phospholipid molecule increases, then cholesterol molecule restrict it. And when at low temperature, when we know without cholesterol, there is a compaction of the phospholipid molecules within the cell membrane, and their mobility will be decreased. But if in presence of cholesterol, the mobility will be increased. Membrane proteins. Integral membrane protein is also called as transmembrane protein or P protein, transmembrane protein, and another is a peripheral protein or extrinsic protein. Uh, they helps in transport of large water soluble and polar molecules. Enzymes like digestive enzymes in small intestines, they are also protein. They are responsible for hydrolysis of different substances like protein, lipid, or disaccharides. Cell membrane protein also act as cytoskeleton. Our glycoprotein and glycolipids are present. Their function is they perform in respiration and photosynthesis. Cell recognition, which can be found in ABO blood group. Receptor molecules or cell signaling, it is found in case of hormones and neurotransmitters transmission. So this is the image showing the structure of the cell membrane. This is the extracellular fluid part, and this is the intracellular fluid. This is the cytoplasm side. So these are the glycoprotein molecules, extrinsic protein. This is the presence of cholesterol. These are the phospholipid bilayer. This is the protein channel, or sodium plus potassium plus pump. This is called as sodium channel. Transport protein, there is transport protein, transmembrane protein also called, you can say. This is the glycolipid structure, and the oxygen and carbon dioxide molecules can so we know we can query, but we can ask why the fluid mosaic model is necessary. Because the oxygen, carbon dioxide, such hydrophilic molecules move throughout the cell in between the phospholipid structures. So fluidity is needed for their transmission, for their transportation of oxygen and carbon dioxide through the cell. This is the phospholipid bilayer. So that brings us to the end of our detailed discussion on cell membrane or plasma membrane. We learned how acts as the protective boundary, follows the fluid mosaic model, and performs vital functions like selective transport, communication, recognition, and maintaining homeostasis. In short, the plasma membrane is truly the gatekeeper of the cell, ensuring life continues smoothly inside every organism. If you found this video helpful, don't forget to like, share, and subscribe to the channel for more detailed and easy to understand biology lessons. Drop your question or suggestion in the comments below. I would love to hear from you. Thank you for watching, and see you in the next class. All the best for your studies. Bye.